Hello everybody. On the our previous video, we have started with introducing you protein databases. Before that, we have introduced you NCBI database to start looking for some information that you need regarding the proteins or other things. So NCBI is known for doing blast searches, alignments, and other things. And an RCSB database where you can download the protein 3D structure and study further. Also, there can be assessed some articles and you can study your protein. Today, we are going to go further, just simply in database, and you can see Uniprot. Yeah, Uniprot is, I would say, bigger database comparing to the RCSB, and it has links with other databases. Here you can see, and also the interesting thing, if in most cases database would have reviewed information, here it would have reviewed part and unreviewed part. So they are not still reviewed, they have information that are not published yet. So that's why it looks more interesting. So as usual, we are going to search for PABP1, for which I'm interested in. So when you search this protein, you can see that here reviewed is 113, unreviewed is 3 times more. Okay, I'm going to look for human PAVP1 and which is reviewed. Yes, let's go to this PABC1. So it is in cytoplasm, Homo sapiens. Okay, what information does it give us? So firstly, it has its unique code for this protein. They're going to first describe the function and have links to other databases, as you can see, some taxonomical information, some other databases. Okay, post-translational modification. I don't know if you have heard about this. It is modifying after translation the protein by adding chemical molecules such as methyl, for example, methyl group, and its methylation. Acetyl group is acetylation. Phosphoryl group, it is phosphorylation. Okay, so these three are more studied than others because there are now many other post-translational modifications. So when your protein have not only the amino acid sequence but also some addition or some cleavage, so it is post-translational modification. So proteomic databases that you can look further, have access here. Each of them have different purpose of study. You can go further. So good thing with the Uniprot, it combines all the information that you need. So here, for example, PTM databases, you can go for the first one, for example, IPTM.net, and look here that it collects all the PTMs, not all. I'm currently studying the post-translational modification of PABP1, and it doesn't have all. I'm trying to collect all the post-translational modification for this particular protein. Okay, let's go further. So you can, if you are interested, you can here study this. Interactions. So what interactions it does have, and it gives you link and you can go and study further. Okay, let's go. Okay, and I think this is the most interesting part. If in the case of RCSB, you should enter for each structure separately. Here, you can study all the structures in one place, and I think it's very good opportunity to have a look which structure do you need, because for example, if you look here, it looks like a big structure and it's from 1 to first, I mean, I see 190, but uh, it appears to be it's repeated parts. So here there's a small part. I think it's C domain in C terminal. Same C terminal with a protein interaction. You should have access to that if you go further. Then also PubC domain with probably other protein interacting here only. So if it is 90, it should be RRM2 probably. Here PubC domain. So and other many other structures that you can look yourself. So it's incredible that you can study this without downloading any uh, other visualizers. For example, for RCSB, you could, but so it's good to have visualizer here. But anyway, I think it's better to download tool such as PyMol or Discovery Studio Visualizer in Chimera. In all cases, they're freely available online. Okay, so here, fast A. So it is a fast A file gives you. Let's download here. You can access this information. It gives you the amino acid sequence for this protein, and you can study different things. Okay, let's just copy this. For example, let's copy this, and if you remember, we have studied NCBI database for blasting, so you can go and do blast. Just search protein to protein. Search if there are proteins with the same sequence, or here different algorithms for doing alignment. So for the reason of the purpose of aligning, you can see many different results for that alignment. So this is a great tool. This is a great database. Without considering other databases, if you are interested in a particular protein, you can do research here and go further. For example, you can simply choose and it goes to other database because it has link, and then it should have information regarding the articles that published and gives you brief information and information that you need regarding the protein. So let's just wait what information they're going to give us. I think probably I have problem with internet access. Okay, so we have result for the PBP1 alignment. Interesting that it has 100% query and 100% identity with hypothetical protein on monodon monoceros macaca mulata PB1. It's really interesting. I didn't know that. I knew that it has 99.6% identification with musculus. However, I didn't know that it have 100% identity. I say just wow. I say wow, really. But our query is has 636. It's PBP1 in human. This protein here is bigger than that. Look. Wow. I say just wow. Okay. So sometimes you can access some unpredicted information. So let's pause just today with here. And as I said, you can access for articles that you want to study here, hundreds of articles written regarding this protein, and whatever you are interested in, you can find here. Hope you enjoyed this video. Thanks for watching. Please do not forget to subscribe. I'm going to further show you what I am going to do with this protein in my next video. See you. Bye.